All right, guys, let's start. Can you hear me in the back? Okay. Okay, so we are continuing studying the various polymer molecules that are present in the living organisms. And so today we're going to actually talk about the next class of molecules called nucleotides and the nucleic acids that are made of the nucleotides. So far you've studied amino acids with, and the proteins with Professor Jeffrey, right? And we talked about sugars last time. So, and we'll talk about the nucleotides and nucleic acids today, and the next lecture would be on the lipids. So, and this would be like perhaps, the four major classes of macromolecules that are present in the biological systems. So this is chapter 8 in the 6th edition of Leininger's Principles of Biochemistry. Okay, so what are the nucleotides and what are their functions? So perhaps I should have started from the very end, actually, of this function list, so that the nucleotides serve as building blocks for nucleic acids. But besides that, they actually play a very important role in the energy metabolism. So you all heard about the ATP, which is like a universal, universal energy currency that is used by all living organisms with only very, very few exceptions. So they can serve as enzyme cofactors. And they also participate in the signal transduction almost as hormones. So, but of course, the main function of the nucleotides is the energy metabolism and building blocks for the nucleic acids. Okay, so if we were to elaborate a little more on this function of nucleotides as a building blocks, so then we will see that they basically used to build the DNA, which is a storage of genetic information. So they used to build the RNA, which can serve either the transmission of the genetic information or processing of some other RNA molecules and so on. Okay, so and also the nucleic acids participate in the protein synthesis, and we'll talk about this in detail in the 4th module of our course. So right now we just sort of mentioned this. Okay. So what are the nucleotides made of? What is their chemical composition? So this group of molecules is a little bit more complicated than the sugars that we studied last time, but the good thing is that there are not so many of them. So sugars, there were plenty of different sugars, right? So, and in terms of nucleotides, of course, there could be also many different nucleotides, but the ones which are present in the biological system, they're relatively few. Okay, so each nucleotide is made basically of the 3 major components. The nitrogenous base, which can be either one of the 2 classes, either purine or pyrimidine. So the sugar, a pentose sugar in this case, it could be either ribose or deoxyribose, and a phosphate. So the phosphate can be mono, di, or triphosphate. So, okay, so the portion of the nucleotide without the phosphate is called a nucleoside. So there's just a difference in the ending. So nucleoside is just the nitrogenous base plus the sugar. So, and well, finally, there could be just, the portion of the nucleoside which comprises only the nuclear base. It's the nuclear base. So here's just a schematic representation of basically of the components of a typical nucleotide. Where we can actually see the base, which is attached to the 1' atom, carbon atom of the ribose. So, and the phosphate, which is always attached to the 5' end. I'm showing you this pyrimidine and purine here only as a source of the numbering. So, so that the atoms numbered like this for pyrimidines and like that for purines. So to me, even now actually, like my research is largely on these molecules, but even now actually I find it a little confusing that they could have started the numbering, for example, like in the case of pyrimidine from the bottom, like 1, 2, 3, 4, 5, 6, and they could have done the same with this, with the purine nucleotides. For whatever reason, actually, according to the formal rules of chemical nomenclature, that has to be numbered that way. So, but all the scientists, even those who sort of implemented that numbering, suffer from that. So I'm not going to be really asking you the numbering of this carbons and nitrogens, I mean, and the nucleotides, but so that you know actually how the numbering is done. Sure. Well, we will get to this in a second. So that would be like, I mean, the almost entire lecture is on that. So we just didn't. Okay, so first let's talk quickly about the nucleobases. Okay, so what are their properties? So as I said, they're derivatives of either pyrimidine or purine, and they're not just made out of carbon, so they contain nitrogen, and they important that that these are aromatic molecules. So which means that they actually have alternating double bonds, and they have planar structure. So as a result of being actually aromatic, so they absorb UV light in the range of the wavelength from 250 to 170 nanometers. By the way, what are the lengths of the visible light? You all had physics, or some of you had physics. Huh?
Approximately 330 to 670 nanometers.
Okay, so 400, 700. So 400 being which color? Blue, and 700? Red, yes. So what is the, okay, so what is the after the red one? So like in the spectrum? Infrared. Okay, so we have infrared on the upper side of the 700, right? And we have UV, ultraviolet, and the lower side. Okay, do we have sensors to actually, I intentionally use the word sensors because we cannot see UV and we cannot see infrared, right? But can we sense it? How? No. Like let's say, just as a human. Huh? Well, I mean, obviously, if we could not, I mean, there would be no point of asking that question. So UV we cannot sense directly. So, but infrared we can actually. You do it all the time. So this is a heat. So perhaps you've seen in the stores, like grocery stores, when they want these rotisserie chickens to kind of be warm, they have these weird lamps. I mean, which make very little light, but a lot of heat actually. So this is infrared. So infrared, I mean, we cannot see it with our eyes, but we can feel it as a heat. Okay, so just a little rehearsal. Okay, pyrimidine nucleobases. So there are three main ones which are present in the nucleic acid. There is cytosine, thymine, and uracil. So the important thing about all of these nucleobases is that they're good H-bond donors and acceptors. And they're neutral at pH 7. So at the bottom here, I'm showing you the structural formulas of all of them. So there is cytosine, which has an amino group in the position 4, and which have the carbonyl group in the position 2. So thymine and uracil, they actually almost the same. So they have two carbonyl groups in the positions 2 and 4. And the only difference between them is that methyl group over here, so on the back. Why am I even saying, for example, cytosine is found in both DNA and RNA, and the pK at N3 is 4.5. Why I am showing you this? Okay, let's look at the cytosine. Okay, what is the N3? N3 is this atom right here. Okay, so pKa 4.5. How do you interpret this data? Like, for example, you had chemistry, and you studied pKa's there. So what does pKa mean? But what the pKa is by definition? No. Okay. So pKa for the given let's say acid-base reaction. So this is a pH, right? At which, by definition, so pKa is a pH at which this particular group half protonated, half deprotonated, like 50-50. So in other words, pKa at N3 of 4.5 means that if the pH would have been 4.5, right, the 50% of this atom would have proton, the other 50 would not have proton, right? But because the actual physiological pH is 3 orders of magnitude higher, like 3 points higher, right? So it's around 7.5. So basically there are 1,000 times less of protons. Don't forget that the pH is a logarithmic scale. So a change of pH by 1 means the amount of protons changes 10 times. So that at pH 7, basically the probability that this atom will be protonated is very close to 0. So that means that at the physiological pH, this middle nitrogen is always deprotonated. So the opposite thing holds true for the thymine and uracil. So it's other way around. So it's protonated. And this is important for the hydrogen bond donor-acceptor properties. So we will see a few slides down the road when we will be talking about the base pairs. So that actually it's very important for this, why Mother Nature chosen these particular nucleobases for being parts or building blocks of the DNA and RNA. Because, I mean, they have this protonated and unprotonated states, so which is important to make the H bonds. Okay, so now we move to the purine nucleobases. There are only two, adenine and guanine. So basically the position which in adenine, it's a position 6, which contains amino group. So in the guanine, it contains carbonyl group, and the amino group is on the carbon 2. So similar to the pyrimidines, these guys are really good hydrogen bond donors and acceptors. So we'll see why. Okay. So because nucleotides are aromatic structures, so they have conjugated basically pi clouds. I mean, so they're flat, so they don't have any chiral atoms, any tetrahedral carbon atoms, or anything like that. Because of that, they do absorb in the UV zone of the spectrum. And basically, the excited states of common nucleobases decay rapidly via the radiationless transitions. So that what basically defines the absorbance in this zone. So here I'm just showing you the absorbance curves of different nucleotides. But what is actually important from that is the sort of the maximum absorbance overall comes at around 260 nanometers. So, and this is basically why nucleic acids such as DNA, RNA have the peak of absorbance at around this wavelength. So when you were studying the amino acids, how do you think which amino acids would be absorbing UV light, just like nucleotides? Yes, tryptophan. Actually, they're already grouped here. Phenylalanine, tyrosine, and tryptophan. Something else? No. Proline, does it have aromatics? Histidine, that's true. Okay. So basically, it's because of the presence of this, because of the presence of these amino acids in the proteins, proteins also do absorb UV light, but they actually, the absorbance maximum of these amino acids altogether comes at around 280 nanometers. So nucleic acids 260, proteins 280. So, and if you work with a protein which does not have any aromatic residues, basically it's a nightmare because it's very hard to measure its concentration since there is no, there are no markers, I mean, that are used for this concentration determination. Okay. Let's very briefly talk about the tautomerism in nucleobases. So what are the tautomers? Tautomers are two isomers of the same molecule, which differs only in the positions of the protons. So basically, it can be exactly the same. It's exactly the same formula. It's just, I mean, that the locations of the protons, I mean, changes. So the most famous are keto-enol tautomerism or lactim-lactam tautomerism. So that's what you probably studied in organic chemistry or inorganic chemistry. So, and the nucleobases, they can also undergo these tautomeric transitions. So they can go this lactim-lactam tautomerism, so this carbonyl can actually turn into hydroxyl and can even turn into the 2 hydroxyls. But that can happen only in solution, but when it's a part of the RNA or DNA molecule, okay, this nitrogen would be involved in a glycosidic bond with the sugar. And as a result, actually, only that first reaction is possible when the nucleotide is a part of the nucleic acid. Okay. Sugars in the nucleotides. Very simple, very easy to remember. There are only 2 possible. Ribose and deoxyribose. So deoxyribose is not even really a sugar because it's a deoxy sugar. So it's so here's the sort of linear formula of ribose, and this is the Haworth projection of the glucose. So the only difference between I'm sorry, not glucose, of course, ribose. So the only difference between ribose and deoxyribose is the presence or absence of this hydroxyl. So ribose has it, deoxyribose has just two hydrogens here. Okay, so how are they connected? So last time when we were talking about, let's say, two monosaccharides basically making a disaccharide or even a polysaccharide, we were mainly talking about O-glycosidic bond, right? So it's when the connection between the two sugars occurs through the oxygen atom. So in the case of nucleotides, the nucleobase is connected to the ribose through the so-called N-glycosidic bond, when the nitrogen of the nucleobase is connected directly to the carbon of the sugar. So the bond is formed between the anomeric carbon of the sugar and in beta configuration. So which means that the terminal, we'll see it in the next slide, that the terminal CH2OH group and the nucleobase look in the same direction. So this N-glycosidic bond, unlike O-glycosidic bond, is actually much more stable toward hydrolysis. Basically, it just, it's important. It may not be so important for the nucleotides themselves, but it's very important and critical for the nucleic acids, right? So which store our genetic information. So if the nucleobases will be falling off like easily, so that would be very hard, if possible at all, to basically maintain it. Okay. And just a couple of words about the phosphates. So the phosphates are negatively charged at neutral pH, and they can be either mono, di, or triphosphate variants of the nucleotides, so which are typically attached to the 5' position. So nucleic acids are built using the 5' 3 phosphates. So either ATP, GTP, TTP, or CTP. So nucleic acids contain one phosphate moiety per nucleotide, and it sometimes, in rare cases, may be attached to other positions. So basically here I'm just showing you that the phosphate which is attached to the 5', by the way, we haven't talked about this numbering, 5'. So the reason why it's called 5', it's to distinguish it from the numbering of carbons or positions in the nucleobase, right? So in the nucleobase, it's just the numbers themselves, like 1, 2, 3, 4, 5. And the numbers in the sugar, in the ribose, actually with this little sign, so which is called prime. So that's why we call it 1', 2', 3', 4', 5', is to distinguish these carbons from the ones which make the nucleobases. Okay, again, so the phosphates in most of the nucleotides is attached to the 5' position. However, it can also be attached to 3', to 2', and even to have like a cycle. So basically, any position which has hydroxyl potentially can accept a phosphate group. Okay, so now we finally get into actually just the side-by-side comparison of all the ribonucleotides, and in the following slide would be the same for deoxyribonucleotides. So here's just, the nucleotide name, which is the entire thing. So adenylate, it's like adenylate acid, basically, or guanylate, or you can also call it adenosine 5' monophosphate. So, and the nucleoside in this case would be called adenosine. So again, nucleoside is a part of the nucleotide which is highlighted by the box here. So, and the nucleotide can be called adenosine 5' monophosphate, or it can be called adenylate, or it can be, we can even use like the single or one-letter codes or three-letter codes. So in the exam, there would be definitely questions in which you will be asked either to draw a formula or to recognize it. So from the picture given. And please pay attention. So when you're actually looking at these molecules, right? So don't just call it adenosine, for example, if it's the one which has phosphate. Or also pay attention to this 2' hydroxyl, whether it's present or not. So, because the deoxyribonucleotides, neither of them has the hydroxyl here, right? So they all have H. So just be attentive. I mean, yeah, the only difference, okay, the only difference between ribonucleotides and deoxyribonucleotides globally is just the presence or absence of this hydroxyl on a 2' carbon. And also, there is only one more difference, is that the deoxyribonucleotide, they don't have U type of nucleotide, they have T, thymine. So it's the one which have the methyl group. So basically, so in ribonucleic acids, I mean, there is uridine, and in deoxyribonucleic acid, there is thymine. But they have exactly the same hydrogen bond capabilities, it's just they have this little tag, I mean, in the form of the methyl group on the back. And again, here's just another way to summarize the nomenclature of the nucleotides. So you have, I mean, monophosphate, it's the nucleobase plus sugar plus 1 phosphate. You can have diphosphate or triphosphate. So, and all of them are equally present, actually. Well, not necessarily equally, but they present in the cytoplasm of the living cells, and they all actively participate in the metabolism. So we cannot say that like triphosphates are this predominant form of the nucleotides, I mean, in the cytoplasm. That's not true, because they always interconvert from ATP, for example, to ADP or even AMP, and so on. So the phosphates are designated as alpha, beta, or gamma. So alpha being the closest to the sugar, and gamma being the like the most distant from the sugar. Okay. And here, just to summarize where they're present. So basically, so the bases such as adenine, guanine, and cytosine are present in both RNA and DNA. So RNA contains uracil as a base, and DNA contains thymine as a base, but these are two interchangeable bases. Okay, how do nucleotides polymerize into nucleic acids? So that happens through the formation of the so-called phosphodiester bonds that link two successive nucleotides in linear polymers. So, and the result of that is nucleic acids. So there are two major types, DNA and RNA. So DNA stands for deoxyribonucleic acids, and RNA stands for ribonucleic acid. So, in the here on the right, I'm just basically showing you, how the nucleotides can be successively polymerized. So one nucleotide, like shown here as an A, can actually, through formation of this phosphodiester bond, be linked to the subsequent nucleotide. So here on the left, I mean, we are looking at the DNA example. So on the right, we're looking at the RNA example. So pay attention to the fact that basically the phosphates in between the nucleotides are attached in between the 3' position and the 5' position. So as a result, each strand of the DNA or RNA always has directionality. So there is a 5' end, so it's where obviously the 5' end of the sugar is located. And there is a 3' end. So, and as a result, all the nucleic acids, all the strands of the nucleic acids are designated as 5' towards 3'. So we'll be using this terminology a lot throughout this course. Okay, so phosphates plus sugars, basically this part, so except for these little boxes showing the individual nucleotides, are called backbone. So because basically they're all the same, regardless of what is the sequence of nucleotide, the backbone part will be always the same. So the only thing which differs is the nucleotides. So in the case of the DNA, the backbone is much more stable than in the case of the RNA. So, and that is largely due to the absence of that hydroxyl, which is very reactive species. So not having these hydroxyls helps making the DNA much more stable. So, and as a result, RNA backbone is, as a result of having this hydroxyl, RNA backbone is unstable. Okay, and this is why it's unstable. So basically what can happen, this hydroxyl on the 2' carbon of the RNA can nucleophilically attack onto the phosphate in between the two consecutive nucleotides. And as a result, the link between these two nucleotides will be lost. See? And the resulting cyclic phosphate will then be, will then be hydrolyzed in turn, but the connection between two consecutive nucleotides will be lost. So, and this is the main reason why the RNA is less stable towards breakage, basically. Okay. We all know that DNA is, we know DNA as a double helix that stores the genetic information. So, however, it wasn't like that. I mean, this knowledge was, we didn't have that knowledge at all times. So it was for the first time in 1868 when Masher first isolated DNA and called it a nuclein. So at the time, nobody even knew what the nuclein is all about, like what is it made of. So it's only not until 1940 that Oswald Avery, Colin MacLeod, and Macklin McCarthy showed that the DNA is a genetic material. So they, and later in 1952, Hershey and Chase removed any remaining doubt about the DNA and not the protein that carries the genetic information. So right now, for us, it's almost like we take it for granted that the DNA is where we have genes. But back at, I mean, there were, for many years, scientists were thinking that the protein is that entity that carries the genetic information. So they were pioneering and extremely important experiments done by Erwin Chargaff. And I intentionally put them, like some of his conclusions here, because I think they're just very neat and interesting in terms of what he found. So he found that the base composition of DNA generally varies from one species to another, which nowadays does not look surprising to us, right? Because obviously the genetic information from an elephant and from a mouse are different. Therefore, they have different genes, and their sequence of DNA is different. Not surprising. But at the time, that was like just the first time discovery. So DNA specimens isolated from different tissues of the same species have the same base composition. So again, not surprising to us now, because the genome in the cell of my eye is the same DNA that would be in my muscle, right? So the base composition of DNA in a given species does not change with an organism, like with age, nutritional state, or change in environment. So that's also what he found. Oops. Which is also not surprising that our genome is pretty much stable throughout our life. I mean, of course, now we do know that it does change, but using the methods that the Erwin Chargaff used, I mean, it was impossible to detect those minor changes. So, and in all cellular DNAs, regardless of the species, the number of adenosine residues is equal to the number of thymine residues. And the same holds true for the G and C. So this perhaps, I mean, is what the Erwin Chargaff is most known for, is for the discovery of the fact that the number of A's always equals to the number of T's, and the number of G's always equals to the number of C's. So, and later, so that actually helped a lot. That was actually this fact, I mean, this discovery was basically the key input for the double helical structure of the DNA. So, and that structure itself is one of, actually, without any exaggeration, so it's one of the most important discoveries in biology. So, and it's important because the structure, well, actually, at the time when they published this paper, which almost transformed the entire science in the world, so they were so humble to write something like, the structure has novel features which are of considerable biological interest. Yeah, so like many decades after, I mean, we have no doubt, I mean, that was the discovery that transformed, I mean, the whole scientific enterprise. The reason why it was so important is because it explained how genetic information can be stored, maintained, and copied. So the copying part was the key here. So, and so who were the players? So there were Rosalind Franklin and Morrison Wilkins. So Morrison Wilkins was the one who was the head of the lab in which the research was going on. So Franklin, Watson, and Crick were just, I mean, the people in the lab. So Rosalind Franklin was the one who collected the diffraction data. and but she was crystallographer who didn't have much background in biology or biochemistry. So, and Maurice Wilkins actually being the principal investigator, so he shared the data with these guys. And after some time, actually, they found a nice and neat explanation of of the data that was collected by Rosalind Franklin. And one of the key pieces of information that they used was that Chargaff's rule, so that adenines always equal to T, and guanines always, G's are always equals to C. So, okay, so and basically here's the result of their discovery. It's the well-known double-stranded model of DNA. So, what are the important features of this model? Well, first of all, so the DNA is made of the two strands of the deoxyribonucleic acid. So you see one strand and you see the other. The strands are antiparallel. So in other words, if one strand goes from 5' direction to 3' direction, then the other strand will be going opposite way. So the 3' will be at the top and the 5' will be at the bottom. So the nucleotides look inwards and they form hydrogen bonds with each other. So A always base pairs with T, and G, or guanines, always pairs with C, or cytidine. So the sugar phosphate backbone is located on the outside, as you can see. Probably the space filling model, I mean, is a better representation to show you that the phosphates are on the outside. Okay. So each of the strands differ in sequence and base composition. So here is just, I mean, a schematic view of this three-dimensional structure. So basically, if we stretch it and put it on the plain paper, so we will see something like this. So we see that each strand of the DNA, it's antiparallel. So this one goes from 5' to 3', and this one is opposite way. So G and C form 3 hydrogen bonds with each other, and A and T forms 2 hydrogen bonds with each other. So, and actually important, okay, this is one force that keeps the two strands together. But what, I mean, there is another very important type of interactions which keeps the structure intact, is the Stekin interaction. So these are van der Waals type of interactions between the adjacent planes of nucleotides. So all of these nucleotides are kind of perpendicular to the long axis of the DNA or RNA molecule. And as a result, so base pairs, they stack one on top of another. And these are hydrophobic interactions, and they actually keep this molecule intact. Okay. Here I'm actually showing you how exactly, I mean, the base pairs look like. So here is on the top there is AT base pair, or it can be the same as AU. The only thing that you need to change is just remove this methyl group from this nucleotide, and you will turn thymine into uridine. So, and here is a G and C. Remember these atoms, I mean, that we talked about, so we talked about their protonation state. So you can see that this middle atom in the case of adenine is never protonated, while the same middle atom in the case of uridine or thymine is always protonated. That is why it's important, because they will always have this capability of forming a hydrogen bond between each other. Also, another hydrogen bond is formed between the amino group and the carbonyl. So carbonyl cannot give protons, it can only accept them. So it's a good acceptor. So an amino group is a good donor of protons because it already has 2. So in a similar way, similar things happen with the GC base pair. So let's actually, here is just a simple example, which probably if you will know how to answer it, I mean, so you understand the material very well. So in samples of DNA isolated from 2 unidentified species of bacteria, X and Y, adenine makes up to 32% and 17% respectively of the total bases. What relative proportions of adenine, guanine, thymine, and cytosine would you expect to find in the 2 DNA samples? How do you approach okay, let's say bacteria X, there is 32% of adenine, yes. Exactly. 64, 100 minus 64, 36, divided by 2, 18. So, you see, I mean, the power of this Chargaff's rule, right? So you know only one, basically the amount of only 1 out of 4 nucleotides, and you can unambiguously detect how many of the other ones should be there in the DNA. Okay, so the same as, I mean, the same logic is for the 17% one, right? So it would be 17% of T, and that would be, so 100 minus 34 would be what, 66? 66 divided by, yeah, divided by 2 would be 33. Okay, now the question is the following. One of the species was isolated from a hot spring, meaning like living at 64 degrees Celsius. So which species of these two is most likely be the thermophilic bacterium, and why? Huh? Why would be the thermophilic? Why? Say it again? Exactly, yeah. So this is actually this is exactly the case. So the bacteria or the organisms that live at higher temperatures, and there are plenty of different bacterial species which can live even in 105 degrees, at 105 degrees Celsius. So this is a temperature higher than the boiling point of water. So, but it happens at a high pressure as well, and at an acidity in which, let's say, if a human will sort of submerge like put a finger in that solution, you will probably see the bone in a few minutes. So, and still, the life can exist at these harsh conditions. So the more, okay, the higher the temperature at which the organism lives, the more GC base pairs would be there in its DNA. And logic is very simple, just to keep it intact, just to keep it together. It's like a long zipper, so that you want to prevent from denaturing. Okay. One of the main consequences of this double-stranded model or double helical model of the DNA was the following. So that's what the Watson and Crick were actually writing in a Nature paper in 1953. So it has not escaped our notice that the specific pairing we have postulated, and they just simply copied it from Chargaff, that we have postulated immediately suggests a possible copying mechanism for the genetic material. Why? Because when you have two strands together, right? So if you separate them, right, you can unambiguously recreate the missing ones for the first and the missing one for the second. So that's what's actually, that's what's depicted on the right here. So if you have like a parent strand, right? So if you sort of denature it or separate the two strands, then by knowing the sequence in one, you can exactly sort of recreate or resynthesize the other. So they were way ahead of that time. So, so at the times when they proposed, I mean, this mechanism of how the replication of DNA could occur. So that was decades before the enzymes such as DNA polymerases or RNA polymerases were discovered and studied. So we will talk about DNA and RNA polymerases in the fourth module of this course. So today we're not going to spend time on that. But that was actually the one another reason why this discovery by Watson and Crick was so powerful and so much transformed the field. So they predicted the existence of the enzymes, and not only that, they predicted the mechanism that the enzyme is going to be using. So, okay. Interestingly, but DNA and RNA can exist in multiple forms. So the form, actually, let me just switch straight to the next slide, and then we'll come back to that one. So the form in which we know the DNA today, it's the so-called B form. So, and that's a normal form in which the DNA exists in the genome in the cell. But there is another form which is called A form. So this is also a double helix. But in that helix, I mean, the planes of the bases are tilted relative to the middle, to the central axis. So, and also that double helix is wider. It's actually wider than the B form, and it has like a hole in the middle if you look from the top. So DNA exists in this form only in the case if it's dried. I mean, so it's not in the aqueous solution, but if it's dry. So the only reason why I wanted to show you that, because this is the main form in which RNA exists in solution. So DNA normally exists in the B form, and RNA, if it's double-stranded, because RNA can have short segments which are double-stranded. It's not like throughout the entire length is double-stranded, but the short segments of RNA that are folded on itself and form the double-stranded structures, I mean, usually exist in the A form. And basically, the only difference between these forms is the conformation of the sugars, the so-called C3 endopucker or C2 endopucker, and the orientation of the base relative to the sugar. So let's go back for a second. So the base can be oriented in two different ways relative to a sugar. It can be in so-called syn conformation, when the nucleobase kind of is rotated towards, I mean, or atop the ribose sugar. Or it can be an anti-conformation in which it rotated away. How do you think, which of these two conformation is the energetically more favorable? Syn or anti? Why? Yeah, that's right. So I would not use steric hindrance because actually it does exist, and when actually this, if I would have like a model of a nucleotide in my hand, so if I simply move that nucleotide on top of the ribose, it will not clash with any atoms on the ribose. But it's a general logics in chemistry that all the atoms do want to get as far away from each other as possible. So, and in this case, basically rotating it into the anti-conformation, so that's a way how it actually can be in a minimum of energy. So, yes? Say it again? Yes, it's a so-called van der Waals. Yeah, it's relatively weak, but it's basically it's repulsion of the atoms from each other. Okay, and there is one more form which is actually very rare. It's so-called Z form. So Z form of DNA, and this is a left-handed. So one thing which I forgot to mention is that the double helix of a DNA or RNA, it's always right-handed. So when you move along it, so it moves that way. So the Z form is a left-handed, and it exists only in the particular G-rich sequences of the DNA. So, and Z stands for zigzag, actually. When I said in the beginning that the DNA and RNA composed only of these four or those four nucleotides, right? So actually that was not entirely true. So there are a number of so-called minor nucleotides and can exist in both DNA and RNA. So here I'm just showing you some that do exist in DNA. So these nucleotides, such as methylcytosine or methyladenosine, so they appear after the DNA or RNA was synthesized. So either like by special enzymes. So there are special enzymes which attach the methyl groups to one atom or to another atom in the nucleotides. So, and these modifications have their own functions. So, for example, in DNA, they largely serve as epigenetic markers. So it's a way to mark own DNA so that the cell can degrade foreign DNA, in the case of bacteria, for example. So in eukaryotes, it's a way to mark which genes should be active and which should be not. So, and could the environment turn genes and on and off and in an inheritable manner. So quite often, for example, I mean, quite often, I mean, when we talk about genetics, right? So genetics is basically the main, I mean, object of studying genetics is a gene, right? And when does the word epigenetics come into play? Is when the, let's say, for example, the genes do not make sense anymore. For example, based on the genes that the organism have, like for example, I have relatively light hair, right? My wife has dark hair. And dark hair, as you know, it's a dominant trait. So, and light hair is the recessive. So basically, my kids have no chance of getting the light hair just because, I mean, if she is a dominant heterozygote. So, but they have light hair just like me. And so this is against genetics, basically. For those of you who took genetics, direct contradiction. How is it possible? So, in these cases, epigenetic word is used to explain. So what does this mean? It means that the genes are not expressed if they have special tags. For example, if the genes from the dominant genes, let's say, of the dark hair, let's say, which my kids get from my wife, were methylated at some positions, they will not be expressed. As a result, it's only my genes were expressed, even though that they are recessive. So, and they shouldn't have been basically kind of shown up. But that happened. So these different methylations of nucleotides in the DNA can serve as such epigenetic marker. So again, here we're not talking about it at all. I'm just sort of mentioning what is it. This takes us to the very important notion and concept in molecular biology called central dogma of molecular biology, which was postulated by Crick, who very early, actually, in his philosophical kind of analysis of the structure of DNA, started to propose that once information has passed into protein, it cannot get out again. In more detail, the transfer of information from nucleic acids to nucleic acid or from nucleic acid to protein may be possible. And indeed, we know that it's possible. But transfer from protein to protein or from protein to nucleic acid is impossible. So that can be very easily or schematically depicted as this. So that the information from DNA can be easily passed to DNA. That's a process of DNA replication. Or from DNA to RNA. That's a process of transcription. So, or from RNA, it can be interpreted into and turned into protein. But once the information got to protein, it cannot go back. So now we can make like a modern day version of the same scheme. See the difference? So you're just adding two arrows. So one arrow is the passing information from RNA to DNA. So that's with the discovery of the retroviruses and the enzyme called reverse transcriptase, which can synthesize DNA using RNA as a template. And with the discovery of some ribozymes which can self-replicate, so and some viruses which contain only the RNA, which can be replicated. Okay. Which can be replicated. So basically, central dogma of molecular biology can be formulated in a very simple way. DNA makes RNA, makes protein, but sort of not back. Okay, this is just a representation of what the central dogma of molecular biology is all about. So we have the DNA in the nucleus, in the chromosome, which contains our genes. Then those genes are not used directly in let's say protein synthesis, that are copied to a temporary carrier such as messenger RNA, and then the messenger RNA is interpreted by this huge machine ribosome, which synthesizes the protein. So this is the same, as the following analogy. So imagine that you have let's say you want to print a photograph from, I don't know, from the Christmas party, right? So when you have it on your computer, obviously you're not taking the whole computer to the CVS store for printing. So, and you're not even taking a hard drive out of the computer to do that. What you do, you copy it on a thumb drive. So, well, now you don't even do that because you have it on your phone, but so you copy it on a thumb drive, and then you go to the CVS store and get it printed. So that big machine which does that printing, so that's actually the ribosome which interprets it. So this slowly takes us to the RNA. So far we talked about the DNA, which is the double-stranded structure whose main function is to store the genetic material. So how the genomes are organized, how many molecules of DNA, what the metabolism, so we'll all study that in the fourth module. So for now, we just need to focus only on the chemical nature of what the DNA is and what the RNA is. So we know that the RNA main difference from the DNA is that it contains ribose instead of deoxyribose, and it contains uracil base instead of a thymine. And here are just the main types of the RNA molecules in a cell. So there are three main types. Messenger RNA, so that's a genetic information carrier. So basically it's a temporary copy of the gene in the chromosome. there is a transfer RNA or tRNA. So this is an adapter molecule, which actually, if we go back to the scheme, right? So the printer that prints your photo, right, prints with ink, right? Ribosome prints with amino acids. So the ink comes in the form of a cartridge. So the amino acid comes being attached to the tRNA. So basically, tRNA or transfer RNA is like a cartridge for the amino acids that are used for printing of proteins. Okay, so the ribosomal RNA, it's the main component of the ribosome. So, and actually, how do you think, which of the three types of the RNA are the most abundant in the cell? Huh? Ribosomal. I heard at least two, maybe more. Yeah, that's right. So ribosomal RNA is the most abundant in the cell. It's actually more than 80% in most cases, because there are many ribosomes. I mean, that are used to synthesize proteins in the cell, and just because of the mass, I mean, like the amount of the nucleotides that the ribosomal RNA contains by mass, this is by far the most abundant. So not necessarily the most abundant in terms of number of molecules, right? But in terms of mass, I mean, that's the most abundant. So also there are microRNAs and small inhibitory RNAs and many other different types of RNAs which are relatively minor in the cells, which have lots of different functions. So messenger RNAs code for polypeptide chains. So the mRNAs are synthesized on DNA template by DNA polymerase. So one mRNA may code for one or several proteins in several proteins in bacterial, and it's usually only for one protein in eukaryotes. So here just to show you that, so in eukaryotes, the messenger RNAs are usually monocystronic. That means that they contain only one gene, so for one protein. But in bacteria, there could be several genes in one mRNA, on one messenger RNA, and so basically they're sequential in this mRNA, so that mRNA can lead to several different proteins. So what's important is that mRNA is usually unstructured, so meaning that it doesn't fold on itself in majority of cases, and it's, I mean, the idea of the mRNA, it needs to be it needs to be untwined, right, so that the ribosome can grab on it and like read it. So it doesn't have any structural components to it. But on the other hand, the tRNA, the transfer RNA, and the ribosomal RNA itself, they have three-dimensional structures. So the RNAs, just like DNA, are capable of forming short segments of the double-stranded RNA. So I'm just showing you here, again, we don't need to know exactly what these molecules are. These are just examples, I mean, to show that the RNA can self-fold into unique three-dimensional structures. And such as, for example, in ribosomal RNA or transfer RNA. And when these unique structures are formed, the non-canonical base pairs and non-canonical hydrogen bond interactions can actually be formed. So, for example, between guanine and uracil. So this is not a Watson-Crick base pair because its geometry is completely different. But this type of interactions is possible in the unique 3D structure. Similar to DNA, there are minor nucleotides in RNA. And again, these nucleotides appear in the RNA not during the synthesis, but after the synthesis, post-transcriptionally. So after the RNA is being synthesized by the RNA polymerase, so these various nucleotides actually have been introduced by the dedicated enzymes. For example, there is a residue called inosine found in the wobble position in the tRNA. So for now, I'm just showing you what that residue looks like. So it looks like the deaminated adenine. Actually, adenine would have an NH2 group here, and this one is just a carbonyl group. So that could be a very interesting base, pseudouridine. Scientists still actually debate about what is its role. So it's actually looks exactly as regular uridine. The only difference is that uridine is attached through the regular N glycosidic bond through the position 1, and this guy is attached through position 5. So it has exactly the same hydrogen bond capabilities and so on, and still it's unclear why does the cell need to actually maintain this huge army of enzymes which introduce hundreds and hundreds of these uridines in human ribosomal RNA, for example. So we don't know that. We don't have an answer. Here's another, actually, here's just a representation of how the transfer RNA looks like. So this is the adapter molecule that matches the amino acid with the codon in the messenger RNA. So, and as I said, unlike mRNA, tRNA, and ribosomal RNA have unique three-dimensional structures. So, and in 3D, transfer RNA looks like a letter L. So it's an L-shaped structure. Basically, it's like a corner. It has one helix going at one direction, and then the other helix is almost perpendicular to the first one. Oops, sorry. So, and what keeps that structure intact, it's not only this, not only the Watson-Crick types of interactions, which are also present there, but also non-canonical long-range interactions in the tRNA, which basically involve 3 nucleotides, sometimes even 4 nucleotides. So, but in every case, there is an intricate network of hydrogen bonds which keeps the unique 3D structure of the given tRNA or ribosomal RNA molecule together. So from here, I would like you to basically remember the, well, actually one of my teachers, the professors who was teaching me molecular biology back many years ago, he get used always to say that, okay, DNA is a stick, meaning it's boring, it's not interesting, it's just a long double-stranded sort of helix, which has the same geometry, like regardless of which part of it you will consider. So, and he was saying that the RNA is a thing, meaning that it's interesting because it has a variety of different structures and shapes. So, and indeed that's true. So that the RNA is very different. So, in the case of DNA, so this Chargaff's rule almost always works, right? Because we're always talking about a double-stranded DNA structure, right? So if you have, let's say, 10% of adenine, you can unambiguously determine what would be the 4 other nucleotides composition. But in the case of the RNA, you cannot do the same because although that the RNA does have some short segments of the, for example, where it forms a double-stranded, let's say where it falls back on itself, right? I mean, therefore there is a complementarity. But these are only for like portion of its length. And as a result, I mean, the same Chargaff rule will not work for the RNA molecule. So if you would be given 10% of adenine, let's say as a composition of any arbitrary RNA molecule, there is no way you can actually know what the other 4 or what the other 3 are. So, and be sort of attentive on the exam. There might be such question. Okay. Now we actually get into a very important property of DNA or RNA molecule in general, the nucleic acids. So it's a thermal denaturation. So here we'll be talking only about the DNA, but the same holds true for the RNA as well. So denaturation can be introduced by high temperature or change in pH. So what the denaturation, basically, it's like separation of the 2 strands. It's like opening the zipper, basically. So it's so this is standard double helical DNA, and the denatured would be the one where the 2 strands are separated one from another. So the reverse process, I mean the process which is reverse to denaturation, is called annealing. It's when basically these 2 strands of DNA that are complementary to each other. So again, complementary doesn't mean the same sequence. So complementary means that in the positions where in one strand there is A, in the other one there will be T, right? So the reverse process is called annealing. So don't miss it with annihilation. So that's a different term. So, okay. During the process of thermal denaturation, covalent bonds remain intact. So in other words, the continuity of each strand is not perturbed. I mean, it's not cut in pieces. So it's just separated. So it's only the hydrogen bonds that are broken. So, and also the base stacking is lost. So because the nucleotides, I mean, which stack nicely in the form when they're in a double-stranded DNA form, upon actually denaturation, upon separation of the strands, now they actually start to randomly rotate, and they do not stack anymore. And if we actually look at the at some properties of the DNA during the denaturation, we can find the like a parameter which is called the melting temperature. So, for example, I mean the graph here. So this is percent of denaturation, and here's actually the temperature. So in one case, I mean, basically the melting temperature is the temperature at which 50% of DNA is denatured. So the same as with the Michaelis constant, remember? So it's a concentration at which the speed of the reaction is 50% of the maximum. So, so can you actually, just by looking at this graph, tell me which one, the blue or red, I mean, have high GC content? The red has what? Exactly. So very simple. So the more GC, the higher the melting, the temperature, and so on. And another question is actually, what is that, like why we can even measure this? What is this denaturation? I mean, it just says denaturation percent. So in what physically we measure? Huh? Say it again? I mean, here hydrogen bonds are still intact, and here they're all broken. So, yeah. But denaturation here, what we measure, we actually look at UV. So the authors of the textbook should have put like UV in the parentheses. So this is absorbance. So why does the absorbance change as we start melting the DNA, start denaturing it? So this is called hyperchromic effect. Basically, you can see that upon denaturation, DNA start to absorb more than it was absorbing when it was like an intact double-stranded helix. So the reason why that happens is, again, let's look back here. The reason why DNA absorbs is because of the aromatic nucleobases, right? So here, these aromatic nucleobases are all parallel to each other and all stacked. So essentially, we're talking about absorbance of the UV light through the entire length of the. You can actually treat that whole DNA molecule as a sort of optic fiber for which the UV light actually is going through and is being absorbed. So what will happen if we will denature it? You see that each individual nucleobase will start kind of roaming almost independently, right? So as a result, the additive absorbance of all of them would be way more than it was when actually they were parallel. Because I mean there was resonance when they're parallel. So let me actually ask you the following question: and how do you think whether the absorbance of the DNA would be higher or lower if we will digest it into individual nucleotides? Huh? Higher, for the exact same reason, because each nucleotide will be free in solution now. Will be not, I mean, its absorbance would be, again, this is not additive. So when you have each nucleotide sort of floating free in solution, and you have, let's say, 1 million of them, so that would be very high absorbance. It would be much higher than if you would have all the same nucleotides lined up nicely in the double-stranded DNA helix. So they will not absorb as anywhere close to the individual ones. Okay, so the hyperchromic effect, basically, it's the increase of the absorbance upon melting of the DNA. Okay. So what are the other factors affecting DNA denaturation? Well, I mean, the major factor which we talked about is the GC content. So, of course, I mean, also the length of the DNA does affect. So the longer DNA segments, I mean, it's harder to denature than the shorter ones. So in the formula for calculating the melting temperature of the given DNA sequence, so it's not only the GC content that is important, but also how long or short is the segment. And pH and ionic strength also important. However, out of the, let's say if we will be talking about the, actually, how do you think, would it be easier to separate DNA at higher or at lower pH? Huh? Would it be easier at acidic or? Why? I mean, mm-hmm.
Detergents will destroy. If you put your finger inside of detergent, it will not. That's the way I'm thinking about it, right?
Yeah, but I mean, let's remember one important thing. So what is the DNA is made of? So just chemical composition. So I'm like bringing actually up this slide from today's lecture. Any hints? Huh? Louder, please. Yeah, so where I want you to look at is here, phosphates. So again, we're talking about the neutral pH. At neutral pH, phosphates are charged, right? So essentially what we have at the neutral pH, the DNA has two wires which are negatively charged next to each other. So they repel each other, so they don't like to be actually next to each other. But because of the lots of hydrogen bonds, so the hydrogen bonds plus the staking interaction, they balance out those repelling of the antiparallel strands of the DNA. So now, if we will start lowering the pH, making it more acidic, what will be happening with these negative phosphates? They will be protonated, right? As a result, the charge will be removed. So that at lower pH, what will be happening is that these wires which were negatively charged will be no more negatively charged, right? So as a result, it would be harder and harder to separate them. So because this repelling of the antiparallel strands of the DNA helping you to actually melt them. So and vice versa. Did you get it or should I get it? Yes. So, okay. So at basic pH, it's easier to separate them at the same temperature, for example. So at the basic pH, it's easier to separate because they would be even more negatively charged, right? As a result, they would repel more, so that you need to have, apply very little effort to get them apart. At the low pH, of course, at the low, if we will be talking about RNA, there are other, aspects come, so that the RNA is just not that stable and so on and so forth. But let's go back where we were. Okay, here. Oops. Okay, so the major factors that affect temperature, of DNA melting are the GC content and its length, and ionic strength and pH in the way that we actually just discussed. So in terms of ionic strength, for example, monovalent ions such as sodium or potassium are not as critical as divalent ions. So the divalent ions like magnesium, for example, which are very important, so they actually bind to those phosphates and work sort of opposite to the pH. So they kind of neutralize them in the phosphates. So denaturation of large DNA molecules is not uniform. So what that means is that when the temperature is being increased, it's not that DNA start to melt everywhere at the same time. So there are zones where it starts melting first, and there are zones where still it stays intact. So as you can guess, these places where it starts melting first are the AT-rich regions. The regions which have predominant, predominant sequences made predominantly out of adenines and thymines. As a result, they have less hydrogen bonds and easier to melt. An important consequence of all these features is the ability of the DNAs to recombine or hybridize. So the sequences of DNA or DNA strands that have that are near or completely complementary, so they can hybridize. And if you have them first at high temperature and then slowly lower the temperature, so they will find each other. Basically, if you have this one side of the strand in one tube, another side of the DNA in the other tube, and if you mix them and slowly cool down, they will hybridize. So hybridization is basically annealing. It's the same as annealing. So it's finding so that each DNA part will find. Each DNA strand will find its partner. So, so some of you who actually work in the labs and do some techniques such as PCR. So this is actually the basis for the PCR. So that hybridization and melting. So the repetitive hybridization and melting is at the base of this extremely important technique, which is nowadays is used everywhere. Okay. As we know that the thing that we afraid the most is the mutation in the DNA. So the alteration in DNA sequence that produce permanent changes in the genetic information are called mutations. Of course, they related to aging and cancer. In other words, as we get older, we accumulate more and more mutations in our genome, and quite often, the reasons why among other reasons, one of the reasons why the cancer progresses is because there is a critical number of the mutations in the particular genes is being accumulated. There are several different types of chemistry actually which is associated with it. For example, deamination. So what that means is that a normal nucleotide cytosine can spontaneously deaminate into uracil. So, and the cell manages to recognize this problem and fix it. How do you think why it can easily recognize that problem in the DNA? Exactly. So because uracil is not a normal part of the DNA, and as a result, it's very easy to sort of invent an enzyme from Mother Nature, right, which will look for the uracils in the DNA and will immediately change them back to cytosine. So that's exactly how it happens. So there could also be depurinizations or other types of okay. If this is not fixed, only then it will turn into mutation. So the other chemical processes that can go with the nucleotides in the DNA that can potentially lead to mutation if they're not fixed. So we'll talk in detail about that in the fourth module. And what is more potent and more dangerous actually in terms of eliciting the mutations in the DNA is the UV light and ionizing radiation and some chemical compounds, are so-called alkylating agents, which directly modify the nucleotides in the DNA, so it is impossible for the cell to repair those changes. Okay, that's it for today.